Okay guys, another short one today for Tuesday. We're going to just do a little review of chapter 1. I'm just going to go over some quick things with you. Make sure you review these things along with the rest of the chapter before the test. And then also don't forget, if you did not finish the review questions from yesterday, make sure you finish them today. So make myself a little bigger here since we don't have a whole bunch on the screen today. So just to talk about what we did in this chapter, we talked about all the levels of organization. Now, in regular bio, we started with an organism. In AP bio, we went all the way from the biosphere to individual atoms. Make sure you know what each one of those levels are. Make sure you know where they break apart from talking about all the species in an area to just talking about the individual species in an area. And then we go down to organism, and so on and so forth, okay? We talked about producers and how producers are going to use light energy. So that's their source of energy for producers, is light, sunlight, obviously. They're going to take that light energy in, they're going to convert it into chemical energy, and then also with the producers are going to fuel a lot of the other organisms. We talked about the example between the producers, plants, and the Thompson gazelle, which eat the plants, and then the cheetahs, which eat the Thompson gazelle. Going back to the Lion King, there's your circle of life right there. We talked about the 3 domains, and you guys can always pause this video. I know I'm talking quick. We talked about the 3 domains. We talked about bacteria, eukarya, and archaea. Difference between them? Well, bacteria and archaea are both going to be prokaryotic organisms. They don't have a nucleus. Instead, they have their DNA just housed in a nucleoid region. And then the eukarya, the last domain, is the only one that is a eukaryotic organism. It has a nucleus in its cell that holds the DNA. Just one more thing about them. We have bacteria and archaea. We just said they're both prokaryotic. They both don't contain a nucleus. The difference between those two, we talked about archaea live in hot environments, and in order to be able to live in these hot environments, their outer cell covering, the cell wall, is actually of a different makeup than the bacterial cells. So that's why they're in 2 different domains, okay? We talked about reductionism, reducing things down to go from like a very broad sense into maybe individual organisms, okay? So we just got our pieces smaller and smaller and smaller. Deductive reasoning and inductive reasoning. If you remember, deductive goes from general to specific, and inductive goes from specific to general. In other words, if we want to talk about, I don't know, dogs, okay? And if you said, oh, look, I have 500 dogs in this area. All these dogs have a tail. My friend, I don't know, Kevin, he has a dog. That dog must have a tail. We're going from very general, because we're talking about all the dogs in this area, 500 dogs, to very specific. We're talking about Kevin's dog, okay? So that's deductive reasoning. Inductive would be like, okay, look, Kevin's dog has a tail. Kevin, his friend, I don't know, Max's dog also has a tail. Oh my goodness, both of these dogs have tails. That means all the dogs in the world probably have a tail. We're going very specific. We're talking about individual dogs. To general, we're talking about the whole population of dogs, okay? And then the last thing in that little section, we talked about emergent properties. These are like combining little things. We did the bicycle example, like combining the wheels and the spokes and all these different things. Bring them all together, and then they serve some kind of purpose. The bicycle can go. After that, guys, we talked about natural selection. Now, in order to have natural selection, in order for evolution to occur and natural selection to be there, we have to have genetic variation. If not, we're not going to have any particular features or adaptations that are advantageous to one group over another group, okay? In order for natural selection to occur, then we need some genetic variation. We need for the organisms to be able to reproduce, obviously. If they can't reproduce, they can't pass on their genes. And then we need those organisms that come about, or the offspring, to have like modified traits. In other words, different traits than some of the parents, okay? And that's how we get evolution. That's how natural selection acts on organisms, and overall, like I said, we get evolution. And with natural selection, guys, we have to have heritable traits, okay? In other words, those traits have to be able to be passed on from one generation to the next generation. So how do we measure natural selection? Well, if one organism is able to reproduce a whole lot and pass on those advantageous traits, then that's how we get more traits in a population, okay? So even if, like, I think there's these bugs called Mayflies. They only live for one day. But if one Mayfly is able to reproduce, 1,000 times, it doesn't matter that it's only living for a day. It's still able to reproduce. If we have another Mayfly that's still living for one day, but it's not able to reproduce, it doesn't matter that it was alive for that day because it's not passing on those traits. We also talked about the whale example with the pelvis. Whales probably at one time had 4 limbs, and then that limb, the back 2 limbs, they went away. Why? Well, we don't know. Something may have happened in the environment that made living in water more advantageous for the whales. So maybe some whales actually had back legs, and at that same time other whales didn't have back legs. Well, the whales without the back legs didn't have to put blood flow to that specific area, so that was good for them. They didn't have to waste as much energy, and that was the advantageous trait. So over time, the whales with the back legs pretty much went extinct. And then the whales without the back legs, they're the ones we see today. For evolution, guys, it is never quick. This whale example probably took, millions of years to come about. So whatever it is, guys, evolution is always gradual. And that's what I'm talking about, the natural process of evolution. You can't take into account like large events, not to bring it up, but if we talk about the Holocaust, okay, they were trying to exterminate one type of person. That's different. You can't bring evolution into those types of events. So natural evolution, that's why I said natural evolution, is always gradual. And then the last thing we talked about, guys, for the chapter was all the scientific method stuff. We talked about the hypotheses and things like that. For a hypothesis, guys, it always has to be testable. You have to be able to test it. And then you're also have to be able to falsify it. So if you do the experiment, you have to say, oh, look, this hypothesis was correct. Or, hey, no, it was not correct. And then after you guys have a hypothesis, it gets tested and all that stuff. What needs to happen is you're not going to be the only one doing it, the experiment. After you come up with certain results, there's going to be scientists all over the world that's going to replicate your experiment, see if they get the same results as you. So in order for your hypothesis to be really good and your experiment to be really good, everything has to be able to be replicated, okay? And then as we went on with the scientific method, we talked about a controlled experiment and a control group and an experimental group. The controlled experiment, we need to have those 2 groups in there, the control group and the experimental group. The experimental group is our reason for doing the whole experiment. That is whatever thing we think is going to change the results. That's our experimental group. The control group is going to show, all right, well, if I know we did the fertilizer example, but if I did something that I know that's going to happen, okay, for instance, the plant growing, I know it's going to grow in regular soil. If it doesn't grow, then hey, okay, something's wrong here. But we can also use that control example to compare to the experimental to see if the experimental group is actually working. Okay, I have my control group and this plant grew 2 centimeters this week. Well, look at my experimental group over here. My goodness, it grew 5 centimeters. That's fantastic, And then you would replicate this with probably, 500 more plants to see if that experimental group is actually working, is making the plant grow faster than the control group. And then lastly, we talked about outliers, guys. I drew the line, our nice line, and then we had 2 outliers there. And that happens in an experiment. We do have some things that go wrong, okay? But when you are, recording your data, you still want to include those outliers in there, but you want to, okay, well, what happened with this outlier? Why did I get this bad result? And see what happened with that group. Well, okay, maybe I put the wrong soil in that plant, and whoops, that wasn't the experimental group, that was a control group, that's why it wasn't growing as much, okay? So even though outliers happen, guys, you have to take into account why, all right? And that's pretty much a basis for the whole chapter, guys. Again, read over everything, read your notes, read your book, go through it one more time before your quiz tomorrow, and we'll have your quiz during normal class time. If you guys want to Zoom very quickly at the beginning of it, I'll be on, and then we can just go right into the quiz. It'll be a Google form. I'll have the link for it in Canvas for you guys. It'll open, and then it will close at the end of class, okay? Also, if you did not submit your Chapter 1 questions, make sure you submit those, guys. All right, have a good day.